Okay, welcome to part 2 of the video lecture for chapter 8. You'll recall at the end of the first part of this, we had talked about, or were talking about, processes that are defined as spontaneous. Okay, trying that black band to go away, sorry, go back. Processes that we called spontaneous. I said a process is spontaneous if it moves forward without an input of energy. So we also talked about the relationship between how much energy a system has, its complexity, its stability, and its entropy. Okay. Now, think about the house we talked about. A brand new house just been built, when you move in, is already breaking down around you. You can't see it because it happens slowly, but if you could stand there on the curb and watch it for 50 or 75 years, you would see it eventually fall in on itself. That's proof positive that it's losing energy. It's a spontaneous process. The house breaking down is spontaneous. So energy-rich, low entropy, complex systems spontaneously break down to energy-poor, simple, high entropy systems. That's the way it works. Your body's giving off heat right now. That's second law. That's your body spontaneously breaking down as energy seeps away from it. The way you fight against that is by consuming food to take in more energy. Okay? All right. So, how does that relate to biological order and disorder? Well, when you look around you in the biological world, you see trillions and trillions and trillions of examples of biological order. All living things are highly ordered, highly structured, complex, and low entropy. The way they are able to stay that way is because they replace ordered forms of matter, food, in other words, ordered forms of matter and energy with less ordered forms. Okay? So if we think about this on one of the largest scales, ecosystems, we can think of an ecosystem as having energy flowing into it through or in the form of sunlight, and that energy eventually exits in the form of heat. The heat that your body is giving off right now used to be sunlight. A plant at some point in the past captured that sunlight, converted it into sugar molecules, and those sugar molecules found their way into your food. You ate them, broke that sugar molecule down, and gave off that energy as heat, and it went right back into the environment again. Okay, so let's talk about biological order and disorder. So you look at these two structures here. First of all, what we have is a sea urchin test or a sea urchin shell on the left, and a sort of cactus plant here on the right. In both cases, you see highly ordered, highly structured, complex entities, right? They are complex structures. They're not random, they're not chaotic. The only way you get that is because the living organisms that they are, in the case of the cactus, and that they were in the case of this sea urchin shell, were able to take in energy, process it, and use it to build that biological structure. You can't build a house without a construction crew showing up and applying energy to construct the house, right? The building materials there on the lot don't spontaneously self-assemble. So this idea of spontaneous and non-spontaneous processes has everything to do with metabolism. Biological order, or the building of it, I should say, is not spontaneous. You have to inject energy into a process in order to build biological order. You have to inject energy into building materials to construct them into a house. It's non-spontaneous. You put energy into it, that means it's non-spontaneous. It won't happen on its own. Think of spontaneous and non-spontaneous in terms of being energetically favorable or not. A spontaneous process is one where a complex system loses energy. That happens on its own. Molecules diffusing from high concentration to low is a complex situation, losing energy and going to chaos. It happens on its own. It's spontaneous. It's energetically favorable. Things always go from high energy to low. That's what spontaneous processes are. That's energetically favorable. High energy to low. Think about water running downhill. That's going from high potential energy, high elevation, to low. It happens automatically without an input of energy. In fact, it releases energy. It's energetically favorable. Now, try and force the water back uphill by pumping it. What do you have to apply? Energy. It won't happen on its own. It's non-spontaneous. It is therefore not energetically favorable. Water doesn't magically run uphill on its own, does it? No, you've never seen that. A house doesn't magically self-assemble. No, you've never seen that. Proteins don't magically self-assemble from amino acids without an input of energy because it's not energetically favorable, right? It's not. Systems in nature don't go from low energy to high on their own. They have to have an input of energy to do that. That's how you should think about this, okay? All right. So to understand these concepts a little bit more in more detail, we have to talk about the concept of free energy. Okay, free energy, or what's known as G, Gibbs free energy. Okay, so free energy is basically the energy that's bound up in a system as potential chemical energy, let's say, okay, in the case of molecules. And biologists want to know which reactions occur spontaneously and which occur only with an input of energy. We want to know which ones are energetically favorable and which ones aren't. To do that, to make that determination, we have to know, or we need to determine rather, the energy changes that occur in chemical reactions, all right? So free energy then is the energy that can do work when temperature and pressure are uniform, as they are in a living cell. You can think of free energy that's potential energy bound up in a sugar molecule, let's say, or a fat molecule that's available for the cell to extract and use to do work. All right, but as we've said, no energy transfer or transformation is 100% efficient, so as a cell breaks that energy out of a sugar molecule, let's say, and uses it to do work, it's going to lose some of that energy as heat. It goes off in the environment as heat. That's second law. We've talked about that. All right, so what we're concerned with here specifically is, or more specifically, is the free energy change, or the change in free energy as we go from one molecule to another, or as we transform or transfer energy from one type or object to another. What we're looking at then is what's called delta G. That triangle there is the Greek letter delta. In chemistry, biology, physics, it means change. So this is delta G as change in free energy. Change in free energy, delta G. We're looking at the energy change as a reaction moves forward. All right. The idea here is that only processes with a negative delta G are spontaneous. So if we can measure the free energy in a system, okay, we can do some math, right? And it's very simple math. We can add and subtract. If we end up with a negative delta G on a reaction, that means the process was spontaneous. That means the reaction went forward on its own without a net input of energy. However, if we have a positive delta G, that means the process, or the reaction rather, was non-spontaneous. It will not go forward without a net input of energy, okay? So negative delta G reactions, spontaneous, they are energetically favorable. That's a house breaking down, water flowing downhill, that kind of thing. Positive delta G processes are non-spontaneous. They are not energetically favorable. These processes require an input of energy. Pumping water uphill, building a house, for example, building a protein out of amino acids, okay? All right, now these spontaneous processes that have a negative delta G, the nice thing about them is that because they give off energy, we go from a high energy reactant to a low energy product, some of that energy that's released as that reaction goes forward can be harnessed to do work. Okay, so free energy here. Free energy is a measure of a system's instability, its tendency to change to a more stable state. I said before that high potential energy systems are unstable, right? Free energy is potential energy. Okay, so during a spontaneous change where a complex system that's energy rich is breaking down to a simpler one that's energy poor, it's becoming more stable as it breaks down. The free energy in that situation decreases, the system loses energy, right? And the stability of the system increases. Okay, equilibrium is a state of maximum stability. Once you have equilibrium, in the case of a cell, that means cell death, all right? Because there's no longer any energy left for the cell to do work. So a process is spontaneous and can do work or perform work only when it is moving toward equilibrium, because that means that spontaneous processes are going forward, molecules are breaking down, energy is being given off, and that energy can be used to do work. But when all the molecules are broken down and there's no more energy to be had, that's equilibrium, the cell will then die, okay, unless it can take in more energy through food or whatever. All right, so let's look at some examples here. So we've talked about the divers on the platform versus in the water. We talked about diffusion in the last chapter, okay, molecules diffusing from high concentration to low. Here we're also now getting ready to talk about complex molecules like sugars breaking down to CO2 and water molecules. All right, so look at these three situations here. They refer to these descriptions, right, these bullet points. In all three of these states, diver on the platform, highly concentrated molecules, or concentration gradient, complex sugar molecule, we have high free energy, higher G. They are unstable situations, and we have a lot of work capacity. Okay, now, diver goes off the platform into the water. Molecules diffuse from high concentration to low. Sugar molecule, or house, breaks down into CO2 and water, or just a pile of rubble, right, depending on the situation. In all three of those situations, we have this going on. These are all spontaneous. They all go this way on their own. We have the free energy of the system decreasing, so our delta G is negative. Here you have high free energy, here you have low. It's going in a negative direction. Think about your bank account. You get paid, you have a lot of money in your checking account. Between now and the next payday, though, what happens to that value of that account? It doesn't go up. Nope, it goes down. So it's going in the minus direction, okay? Delta G negative. Energy is being lost. Okay, the system becomes more stable. Diver in the water is more stable than up on the platform. These molecules diffused where the concentration gradient no longer exists. They'll stay that way forever, right? These molecules will stay this way forever unless they're rebuilt back into a sugar. These will stay this way unless they're reconcentrated back into a highly concentrated area, right? What does the swimmer have to do to get back up on the platform once he's in the water? Apply energy to move back up on the platform. So the released free energy in all these cases, as we go from here to here, here to here, here to here, can be harnessed to do work. But then we end up with the diver in the water, molecules evenly distributed, no more gradient, and CO2 and water. These are low free energy systems. They are more stable and little or no work capacity. So in every case, we're going from high energy, low entropy, unstable situations to low energy, high entropy, stable situations, right? So going in this direction is spontaneous. If you want to go the opposite direction, what do you have to apply? Energy, non-spontaneous. Okay. All right, if you remember those concepts, it will help you a lot throughout this chapter and the next couple of chapters. Okay, so before we talked about catabolic and anabolic reactions. Catabolic and anabolic really refer to the structure, what's happening to the structure in the process of those reactions. In catabolic reactions, you have complex molecules breaking down to simple molecules. Anabolic reactions are the reverse, simple molecules being built into complex. Now, two more terms, the first of which you see here, exergonic and endergonic. These basically describe catabolic and anabolic reactions, but they're doing so based on what's happening to the energy content, okay, energy transfer. Okay, so first one is the exergonic reaction. In an exergonic reaction, we have energy being released. So these are spontaneous processes. Catabolic reactions, when they occur, they release energy, so they can also be referred to as exergonic. Let's take a look. On this graph, we have our progress of a reaction from beginning to end on the x-axis, and the amount of free energy or potential energy available. You see that in a catabolic reaction, that's also exergonic, we start off with reactants that are high free energy, like glucose, for example, in cell respiration. As the reaction goes forward, those reactants break down into low energy products. So this is the house breaking down, the diver coming off the platform, molecules diffusing from high concentration to low. You get the idea. In every case, it's spontaneous because energy is given off to the environment. That's why we call them exergonic. Exer, exterior, right? Okay, so let's look at the delta G, the change in free energy. So what's the difference in the energy content, the free energy of our reactants and our products? It's this difference here. So we've gone from high energy reactants to low energy products. Our delta G has been negative. It's less than 0, right? It's going in the minus direction. It's decreasing as the reaction goes forward. That's why our delta G is negative. Whenever you see a negative delta G, less than 0, you know that that chemical reaction or process was exergonic and spontaneous, energetically favorable, all right? Now, let's look at endergonic. Ender, inside, interior. Okay, think endo, interior. An endergonic reaction is also an anabolic reaction. Remember, anabolic reactions build, right? So like building the house, that's an anabolic process. We've already said, because it's not energetically favorable, it's not spontaneous, the building materials won't self-assemble into a house without a construction crew. The water won't flow uphill on its own, right? Okay, so endergonic reactions are anabolic. We have low energy reactants, think photosynthesis, CO2 and water. We need sunlight, energy in the form of sunlight, to build them into high energy products, sugar molecules, right? Okay, that doesn't happen on its own. You've got to have an input of energy into the process. How much energy? That's our delta G. It's the difference between our low energy products and our low energy reactants and high energy products. In this case, energy has to go up, so our delta G is positive, it's greater than 0. So if I show you a chemical reaction and I give you the delta G as a number, it's quantifying or measuring the energy change, and it's positive, right there off the bat you know it's endergonic, anabolic, non-spontaneous, and energetically unfavorable. It won't happen on its own. You've got to have an input of energy into it. Okay? All right. So let's look at some examples here with some analogies. So let's imagine that we have this sort of hydroelectric system here, right? We have two vessels of water, one at high elevation, one at low. They're connected by a tunnel that allows water to flow. And because we know water flows downhill, that's what's happening here. We have a turbine in between them. As the water goes from high elevation to low, it spins the turbine. That actually converts the, go back for a second, we have high potential energy here, water at high elevation, low there. That potential is converted to kinetic of water flow. That kinetic energy is then converted by the turbine into electricity. That's exactly how a hydroelectric power dam works, right? Think about the Hoover Dam on Lake Mead in Nevada. That powers much of the southwestern United States because water flows through the dam, converting potential to kinetic and kinetic to electricity. But here we have a delta G less than 0. Water will flow downhill if you give it an opening, right? So it happens on its own, it's spontaneous. And as it does so, it gives off energy. What's the proof of that? The light comes on. All right, that's our delta G less than 0 in an exergonic, catabolic, spontaneous process. But what happens when the water level on this side matches that on the left? That's equilibrium. The water stops flowing, turbine stops spinning, light goes out, delta G equals 0. No longer an energy transformation. For a cell, that's death, right? Okay, our bodies are always working or moving toward equilibrium, and the way that we stave that off from happening is by eating more food. Okay, so cells are not, living cells anyway, are not in equilibrium. They're open systems experiencing a constant flow of materials and energy in and out. This is a defining feature of life. Metabolism is never at equilibrium, right? If it goes to equilibrium, that's cell death. So a catabolic pathway then in a cell releases free energy in a series of reactions. Here you see one reaction, so this doesn't really explain it very well, but it's showing you the concept. But if you look at it in terms of a catabolic reaction, this is more like what you have. So let's take a look at panel A up here first, right? This is an open system. So here we have the same system we had before, but notice that we have an outlet now. So water can come out of the vessel on the right, but we have an inflow of water on the vessel on the left. That maintains a higher elevation here and a lower elevation there, maintains higher potential energy on the left, lower on the right. That system is not at equilibrium, and it will stay that way as long as the water continues to come in, as long as there's an input of water. In the analogy of the cell, this means an input of energy. As long as we have that process going on, our light stays on. Delta G remains negative. Now, B here is a better analogy for a multi-step chemical pathway. So here we have our initial reaction right here, converting energy in one form to kinetic and then electricity, right? Now we get down to this vessel, but we now have this vessel connected to another one. So there's another reaction here that can take this potential energy and use it, right? So the light comes on again through our second reaction. There's still energy left in this one because it's higher than this final one. We can still take out some more energy. So at every step of the way, our delta G is negative, and eventually you get to a point where there's just not enough energy left in your products to do anything. This is a great analogy for cell respiration, which we'll see in chapter 9. Complex sugar molecule broken down over a series of steps, ultimately into CO2 and water, and each step of the way, energy is siphoned off and used to do work. All right, so let's talk about the kinds of work that a cell can do by extracting energy from food molecules through these multi-step exergonic catabolic reactions. Well, a cell can do chemical work. We'll talk about what that is here in a little bit. It can also do transport work, and it can do mechanical work, all right? We're going to see examples of all of these in a little bit, so don't worry about exactly what they are right now. But to do these three types of work, a cell must be able to carry out a process known as energy coupling. Okay, energy coupling. This means using an exergonic process to drive an endergonic process. So extracting energy through an exergonic process and then applying that energy to an endergonic process to make it go forward. All right, this is where we get into talking about ATP, or adenosine triphosphate. Oops. Adenosine triphosphate, okay, ATP. ATP is the middleman that helps our cells to link an exergonic reaction to an endergonic reaction, and therefore allows energy coupling to take place. All right, let's look at ATP a little bit. So there you see it, ATP, adenosine triphosphate. Just think of this as the molecule that carries energy around cells, okay? ATP is composed of really three parts. You have a sugar, all right? You have a nitrogenous base and three phosphate groups. Okay, so the nitrogenous base portion is right here, the sugar is here, and you have 1, 2, 3 phosphate groups. These phosphate groups represent the potential energy of that molecule. They're like the fuel tank, essentially, okay? So you see it there in this space-filling model. This is it again in a ball and stick model, but you can see that it's relatively simple. And if you think about it, if you go back to chapter 5 where we talked about nucleotides, which are the building blocks of DNA, I said every nucleotide has three parts. It has a nitrogenous base, a sugar. In the case of DNA, it's deoxyribose. In the case of RNA, it's ribose, right? Ribose. And it has one phosphate group. So every nucleotide has a base, a sugar, and a phosphate. Well, ATP here has a base, in this case, adenine. It has a sugar, ribose, and not one, but three phosphates. So ATP is a modified nucleic acid nucleotide. It's a modified monomer building block of RNA specifically, because the sugar is ribose, all right? Modified in the sense it has two extra phosphate groups, and these three phosphate groups, or the covalent bonds between them, represent the fuel tank. This is where the energy of the molecule is stored. And because ATP has so much energy in it, you got to realize it's extremely unstable. It's complex, it's unstable, it's low entropy. If it's unstable because it has so much energy in it, it very easily breaks down into a more stable state. It does that by splitting off phosphate groups. We saw that when we were talking about the sodium potassium pump in chapter 5, for example. Okay, I'm sorry, chapter 7. All right, how it donates phosphate groups to other molecules and therefore injects energy into them. So ATP is energy rich and very unstable. So let's take a look. So this is the structure of the molecule. Okay, so again, you see our adenine base, ribose is the sugar, and then 3 phosphate groups. Okay, so what you see here is an exergonic process whereby the ATP molecule donates a phosphate. The first phosphate to come off is that third one there. It breaks away, and now we refer to it as an inorganic phosphate because phosphate groups are just PO4. There's no carbon, no hydrogen, they're organic. But while they're attached to this organic molecule, they're considered to be in organic form. Break that bond, separate it, now it's inorganic. That's why that subscript I is there. Okay, when that happens, it's catabolic and exergonic, energy is released. A lot of energy is released. We had adenosine triphosphate, 3 phosphates, break it down to a more stable state, adenosine diphosphate, 2 phosphates. Energy is given away or being given off. The delta G here, as that reaction goes forward, is negative. Okay? All right. That is ATP hydrolysis. Notice using a water molecule like we talked about, right? ATP hydrolysis to separate that phosphate. And a good bit of this energy that's given off can be used to drive some anabolic process. All right. So the bonds between the phosphate groups of ATP's tail are broken by hydrolysis. We talked about that. Energy is released when the ATP from the terminal phosphate, energy is released from ATP when the terminal phosphate bond is broken, and that release of energy comes from the chemical change to a state of lower free energy. We go from higher free energy to lower free energy, energy is released. Diver comes off the platform, energy is released. Molecules diffuse from high concentration to low, energy is released. Sugar molecule breaks down to CO2 and water, energy is released. House breaks down or burns down and becomes a pile of smoldering rubble, energy is released. It's all the same thing. They're all exergonic catabolic processes with negative delta G, spontaneous and energetically favorable. Okay, so how does the hydrolysis of ATP by splitting off phosphates perform work? Well, we said we had 3 types of work, right? Mechanical, transport, and chemical. All of them are powered by hydrolysis of ATP. This energy given off from ATP when it breaks down like that powers all 3 types of work. So, in the cell, the energy from the exergonic reaction of ATP hydrolysis that you see here can be used to drive an endergonic reaction. When you consider the 2 reactions together, overall, the coupled reactions are exergonic, and any exergonic process will move forward because it's energetically favorable, okay? So, ATP will drive endergonic reactions by phosphorylation, transferring a phosphate group to some other molecule, some reactant. That reactant molecule that has a phosphate group from ATP now attached to it is called a phosphorylated intermediate. Okay, write that down. A phosphorylated intermediate. So, when I talked about the sodium potassium pump having a phosphate group from ATP attached to it, allowing it to do work, that protein, sodium potassium pump, was then a phosphorylated intermediate. Anytime a phosphate from ATP is attached to another molecule, that ATP is injecting energy into that molecule, and that molecule is therefore called a phosphorylated intermediate. All right, when you inject energy into any structure, house, protein, whatever, you are making it less stable. You always have to remember that. High energy systems are less stable than low energy systems. So if you add energy to a system, you're making it more unstable, right? Okay. You got to realize that or understand that in order to understand this next concept that we're going to talk about. Okay, so let's look at how it is that energy coupling works. So here we have a chemical reaction where something called glutamic acid is going to react with ammonia, and that makes a product called glutamine. Glutamine is an amino acid. It's one of the 20 that are used to build proteins, right? So we're building the house here, right? We're taking building materials, glutamic acid and ammonia, and constructing them into something more complex. It's like building the house. So if you're constructing, you got to know this is anabolic and endergonic. So delta G is positive. So if you want to make a mole of glutamine, 6.02 times 10 to the 23rd copies of it, you need 3.4 thousand calories to do it. That's the amount of energy it needs to take. It needs to build 1 mole of glutamine. Okay? All right. So that's not going to happen without an input of energy. That's like water running uphill, a house building itself, all that, right? It's non-spontaneous, not energetically favorable. Okay, how do we convert it into a spontaneous process that is energetically favorable? Excuse me. We do that with ATP. So if we take glutamic acid and add ATP to the mix, ATP phosphorylates it, third phosphate splits off ATP, and now you see we have a phosphorylated intermediate, right? Okay, that phosphorylated intermediate is now very unstable because this phosphate represents an injection of energy into it. Systems in nature don't like being unstable. They always go from unstable to stable. So what happens next is the ammonia comes in, replaces the phosphate, and attaches to glutamic acid, making glutamine. That's now a more stable state. So we went from stable to unstable and now back to stable again, okay? Notice also that when the phosphate came off of ATP, we're left with ADP, adenosine diphosphate, okay? So we've gone from highly unstable, energy rich, to more stable, more energy poor. Where'd the energy go? It was injected into glutamic acid through the phosphate, the phosphorylation, which then made this, a phosphorylated intermediate, very, very unstable. It also wants to go back and become more stable. Here, how does it do that? The ammonia replaces the phosphate and it becomes glutamine, which is more stable than phosphorylated glutamic acid. And you end up with ADP now plus inorganic phosphate. All right, so if we want to understand why this works, we have to think about how much energy is released from the ATP when it splits off the phosphate. In other words, how much energy does ATP hydrolysis liberate or produce that can then be used to do work? So if you look down here, we know that to build a mole of glutamine, we need 3.4 thousand calories. It's endergonic, non-spontaneous, positive delta G. Now, what about breaking down a mole of ATP? 6.02 times 10 to the 23rd copies of it. If you break down all those, you get out 7.3 thousand calories. That is more than twice as much energy as we need to make a mole of glutamine. So this is our endergonic anabolic process. That is our exergonic catabolic process. They're linked by ATP in the middle, and we end up with a negative delta G. Plus 3.4 thousand calories minus 7.3 thousand calories gives us overall a negative delta G. So more energy has been given off than was used to build glutamine, and so we have excess energy going off in the environment. So we've converted an endergonic non-spontaneous process up here, building glutamine, into an exergonic spontaneous process by supplying energy from ATP. We've forced this what was anabolic, endergonic, non-spontaneous process to become spontaneous by throwing ATP into the mix. All right, that's how ATP drives chemical reactions forward. The energy from the ATP was used to energize the system and make it move forward. Okay? All right, and that's what we do anytime we do work. The work won't happen on its own. We have to apply energy to make it happen, so we're converting an anabolic process, a non-spontaneous process, to a catabolic, I should say exergonic spontaneous process. Okay? You're converting an endergonic process, which is this first reaction, into an exergonic process. How you know it's exergonic? Because you see that negative delta G. All right, so this is an example of chemical work, right? The first category of work that ATP hydrolysis allows to take place. Okay, but transport and mechanical work are also powered by ATP hydrolysis. And this usually works, think about transport work. We talked about vesicles being moved by a transport protein from the endoplasmic reticulum down to the Golgi, for example, walking along those little microtubule fibers, right? That protein that's doing that work has a subtle change in shape as ATP phosphorylates it, and that's what causes it to sort of march down that microtubule and carry that vesicle, okay? So let's take a look at some other examples. Here we have another example of transport work. Not in this case, we have an active transport protein that you see right here, pumping a solute from low concentration back to high. Think sodium-potassium pump or proton pump of the plant. There you see a phosphorylated protein. That's a phosphorylated intermediate. That phosphate is giving that protein pump the energy it needs to move something against its concentration gradient. That is endergonic and non-spontaneous. How do we make it happen? We add energy to force it to go forward, okay? And you see, you end up with ADP and phosphate as byproducts. Here we have the motor protein I mentioned, right, carrying the vesicle. So walking this way, let's say. Every time a little leg moves forward, that's a phosphate from ATP driving that work. So breaking down ATP to move that vesicle from point A to point B. The first one's transport work, the second one's mechanical work. We also talked about chemical work in the case of making glutamine from glutamic acid. All right, so all of these things should tell you that we're burning through our ATP very, very quickly. On average, a typical cell burns through about 10 million molecules of ATP per second. That means we have to regenerate it very quickly. We have to remake more ATP. And good thing is that ATP is a renewable resource. It's regenerated by the addition of phosphate groups back onto ADP to make more ATP, all right? So if we want to add that phosphate back onto ADP to build it back into ATP, what do you need? Well, this is anabolic, it's endergonic, non-spontaneous. To build it back into ATP, you need a source of energy. So we have what's called the ATP cycle. This is sort of like a metabolic revolving door through which energy passes during its transfer from catabolic to anabolic pathways. Let's take a look. So here we have the ATP cycle, right? So notice we have ADP and phosphate on the bottom, representing low energy. To build that back up into ATP, that's like building the house. It's like building glutamine from glutamic acid. It's like water running uphill, right? In every case, you need a supply of energy coming in. Where does this energy come from? Catabolism. Catabolic reactions that, in the case of the cell, break down food molecules to supply energy. So these are exergonic, exergonic spontaneous processes, energetically favorable. Proteins, fats, sugars, whatever break down. They supply energy to the cell through cell respiration, which then builds ADP and phosphate back into ATP. Now the ATP goes through ATP hydrolysis, splitting off phosphates that then power endergonic, non-spontaneous, energy-consuming processes. Mechanical work, chemical work, transport work, all right? Now, take the energy back out of ATP by phosphorylation. What do you get? ADP plus inorganic phosphate again, which has to be rebuilt back into more ATP. Energy from food molecules provides or helps that to happen. That's the ATP cycle, okay? All right. I think this is a good place to stop for the second video. Hopefully I can get everything, the rest of this done in the third video, but we're going to start up talking about enzymes next.